Okay, Germany, and postdoctoral experience in the Indian Institute of Science, Bangalore, and the Finnish Museum of Natural History, Helsinki. She currently heads the research collections at the NCBS, and where she curates the Lepidoptera specimens. Her work explores biodiversity, species traits, and ecological interactions by integrating field and museum research. Passionate about public engagement, she promotes citizen science and coordinates National Moth Week in India to make natural history accessible to all. Pritha.
It's on. Hello, good morning, everyone. My name is Pritha. Step away from the. How do I change the slides? Or do it like this then? Okay. Yeah. Okay. Okay. All right. So I'll just start off with the talk. Uh, First disclaimer, I don't know Malayalam at all. Okay, so if you need any translation, my friends will help me. I will do the entire talk in English. So if you have any questions, please feel free to stop me and ask. I'm happy to answer in between the talk as well. So, okay, we'll start with what are natural history collections. So, these are curated biological collections. These could be geological specimens also, like rocks, minerals, fossils. They can also be anthropological materials. But when we talk of just natural history collections, we talk of biological specimens. Uh, all like plants, animals, fungi, microbes. So all the bi- yeah. So okay, so why are they important? Natural history collections provides a historical record. So it is more than just a stationary collection. It is more for the future. So whatever we are collecting now, we are preserving them in our specimen collections. It helps future research. It might not be useful the very day that it is collected and stored and preserved, but maybe 10 years later somebody comes and studies those specimens, they can access those, right? So and they form a baseline data for environmental and evolutionary research. And also in the era when we are losing more species than we know more, like new species are there, which like we are losing more species than we are discovering new species. So in this context, natural history collections are a very useful resource, because if you find something new, what do you compare it with? Like you can go back to these collections, look at the type specimens, and maybe confirm whether it's a new species to science or not. But how are these specimens collected? Do we just go out and see something interesting and collect them? No. When you collect a plant or animal, insect, bird, whatever, you need to know what exactly to do with this specimen. It's I mean, we don't encourage rampant collection. Like just because you're curious what this is, you collect and you keep it with you. No, you need to understand that this might be useful. This might be useful for not only for yourself, but for somebody else who accesses them and compares it with other taxa, other species, or whatever, and use it in some way, useful for science. So we should be responsible about that as well. Okay. So, okay, and how it is done? It is done through field work. The researchers who are well versed with the field of taxonomy, the field of natural history. They go out, they use different methods for different taxa, and they collect them. And not only the specimen is collected, but you have the ecological data as well, from where it is collected, the date, the habitat type, and other ecological notes. And all of this are done through proper forest department permits. You need permits to ensure that these are sustainable and legally compliant, and there has to be some ethical understanding to minimize environmental disruption and also respect local communities. But what happens when a specimen comes into a museum? So these are labeled. They are quality control. We look for quality control. We make sure that they are in a good condition to be preserved for the next some like 100 years, so that people can come back and look at them and use them as a useful resource. And they have all the data attached to them, like species name, collector's name, date, location, habitat, etc., whatever can be gathered. So it is more than just a specimen, so it has a story behind it. So if you're collecting an insect, a moth from Wayanad, and you're taking it to Bangalore, it has all the ecological information with it as well, and not just the specimen. And yes, we maintain a database where all these information can be stored, and also make it open access. So there are many platforms, like I would like for in the birding parlance, eBird, something like eBird. Like we can also have something like that for each taxa, and also there is something called as a global biodiversity information facility, where all these data can be made open access. So, like, why are these collections becoming more and more important in the Anthropocene? So, the era that we live in is termed by evolutionary biologists as the Anthropocene. Okay, it's a debatable topic about the term, but yes, humans are having more of an impact on the environment nowadays. It's more visible how humans are impacting the environment around us. And in this era of rampant degradation, biological collections are becoming more and more important, because they have the data from the past. Maybe the species is gone, but you don't see it anymore, but the collections have them. You can use them as repositories for these kind of data, for our knowledge that how anthropogenic drivers are affecting biodiversity and natural resources. And with the changing global change is a very hot topic for research nowadays. And with global change, the interactions of different species are also changing. So it's useful for like the next generation can use these collections for like different methods they can use, like citizen science, more advanced analytical techniques, automation, and all of these can be used on museum specimens to answer ecological and evolutionary questions. So basically, naturalistic collections lie at the nexus of all these fields, like systematics, ecology, conservation, applied research, public health, habitat loss, global change, etc. And there's also a human dimension to it as well. So when you see a box of, for example, so I've shown her butterflies. I am a very insect biased person, but since we are here in a bird festival, I can imagine this is a box of bird specimens. Like, what are the kind of questions that you can answer? Like, you can, for your research, you can talk about biodiversity and systematics. You can talk about adaptation and to environmental change, how the population is shifting, and again species interaction, evolutionary interactions. And with the use of advanced techniques like museomics and ancient DNA, you can also study these specimens. Like I said, that with the advancement of technology, you have techniques like micro CT, which is a technique for creating 3D images, high resolution 3D images. You can look inside the specimens. You don't really have to damage the specimens, and to understand what's inside it, you use micro CT and look inside it if you want to study the anatomy in detail. So all these techniques can be used. Genetics, you can use tissues from different specimens and use them to answer genomics related or other DNA related questions. And you can also use other measurement techniques, for morphometric techniques, to look at specimens and compare their, for example, wing morphology of different dragonflies. So different techniques can be used to study these specimens. So I'll just give you some example from different taxa before I move into birds. So for example, these bumblebees, they carry pollens on their legs. So if you do, for example, DNA analysis of those pollens from a bumblebee which was found 100 years ago in a place, and you compare it with a bumblebee from the present day, you can actually see if there is a shift in their diet. You know, you can like how, and like degrading environment or changing environment is also affecting their diet, and all of those things you can study from pollens found on the bumblebee specimens. And also to study evolution, for example, the entire Himalayan ecosystem. So if we go back into time, how was Himalaya formed? So there was, there was a like India, it banged into the larger Asia continent, and the Himalaya mountains, they emerged. It's a very young mountain system. With that biogeographic information, you can also see how species which live in the entire Himalayan belt have evolved. For example, here our collaborators from the Thakre Wildlife Foundation, they studied these gecko species from the genus Certodactylus, and they looked at species from throughout the Himalaya and traced back their phylogeny using molecular and morphological techniques, and they found that as they relate how Himalayas were formed, they could also understand how this genus of gecko also evolved. So you can relate species evolution to biogeography as well. And this study actually led to discovering new species in this genus, Certodactylus. So yeah, so one question leads to another using just museum specimens. And this is something that it's part of my study. So what it is just to highlight the method of micro CT, where I was mentioning that you can look inside the specimens without harming them. So I looked at museum specimens from India and in Finland, and I wanted to look inside the moth and study their hearing organs. So none of the specimens were damaged, and we did micro CT to look into the hearing organ. I don't have a pointer here, but this yellow structure that you see in the abdomen, it's actually the hearing organ of the moth. And yeah, so it's a 3D structure. So it kind of looks like this. So you can make these cool 3D videos of different organs inside the specimen. Imagine if you want to study inside of a bird, for example, you can use this technique and look at the anatomy to answer different kinds of questions. Okay, so so we have to talk about birds here, and birds are also, like, there's so many studies which have used museum specimens to answer macroecological questions. For example, feathers of the birds are very, very important. If you look at a bird specimen, it has different kinds of feathers. Some are flight feathers, some are feathers used for thermoregulation. So each kind of feather has a function, and if you study a specimen, and so we have a project which I'll talk about later in NCBS called the Feather Library, which documents the different kinds of feathers in different birds species found in India. Just by looking at the feathers, you can tell a lot about how the bird is surviving out in the wild or not. Maybe it's an extinct bird. You can look at those specimens and try and understand these kind of questions as well. So this study was done in the Himalayas by Sahas Barve and his team. He's Indian origin ornithologist, but he's working in the US. So he looked at about 250 specimens of birds across Himalaya from different elevation, and he just wanted to understand how these birds are adapting to the colder environments or not, and how does size of the bird also help them adapt to these environments. So he looked at the feathers, and all these specimens he studied in the Smithsonian in the US. And he studied the section of the feathers called the downy section, as you can see, and there's also a pennaceous section. And he concluded that increase in the proportion of the feathers downy section helps birds to adapt at a colder temperature, and that is common across tropical as well as temperate species. And smaller bodied birds tend to have longer and more insulative feathers. This entire work, this is a fantastic piece of work done across Himalayas, was done based on museum specimens. Then we come to another study which looked at barbets by Dr. Anand Krishnan and Krishna Priyatamma. It looked at acoustic traits, like what kind of songs these barbets are producing, as well as their morphological traits. And they found that in sympatric communities, in the same communities, the traits of the morphological traits and the acoustic traits are divergent. This was also based entirely looking at museum specimens. And a more recent study that came out probably last month, if I'm not completely wrong, Vijay Ramesh and team, they looked at grassland bird species, and they looked at their species decline with colonial era landscape change in a tropical mountain ecosystem. This is a very, very interesting study in my opinion, because it not only used museum specimens, it also used historical data. I think Parvati will talk about how historical data is also important for bird research. So this study integrated both museum and historical data to unearth how these changes in the landscape has caused decline in this grassland bird species. So more than just answering ecological questions, evolutionary questions, these bird collections are also useful for rare bird specimens. So in our collection at NCBS, as part of the feather library, we have these two specimens of the white tern and the sooty shearwater, which are rare bird specimens, and you can access them from our collections if you are interested to do some study on them. And also in NCBS we have the scanning electron microscope facility, which is also a technique. You can look at a very minor scale, like you can zoom into the bird feathers and look into their structures by using this technique. So our collaborator Esha Munshi, who runs the feather library, is also working on these things. So yeah, so it's just not a specimen. You can do so many things by using these specimens and looking into the feather structures, the ultra structures of feathers. You can tell a lot about flight mechanics, thermoregulation, coloration, and how they are adapting to the changes in the environment. So as I was speaking about this project, Feather Library, we have about more than 200 curated specimens and many many uncurated specimens uh, in NCBS, which is led by Esha Munshi. She's our collaborator, and it's a very very important project nowadays because in India you cannot really collect bird specimens, but this project allows it to collect dead birds. We collaborate with rescue centers and the forest department, and from where we get dead birds, and we are building this collection so as to facilitate ecological and evolutionary research. So do check out, if you are interested, the Feather Library website. It has more information on this. So yeah, just to wrap up, like this is what I was trying to tell, that natural history collections are very dynamic. They look like static collections of dead animals and plants, but they are a dynamic resource, and they lie at an intersection of science, educational outreach, policy initiatives, and teaching and research. And recently, some of us, not some of, many of us, we collaborated to bring out this information in form of this paper, which was published 2 weeks ago. What So we have a lot of collections in different institutes in India, but we don't have a nationalized center for biodiversity research, which we want to propose. Is that, and so that it is easily accessible to for ecologists and young researchers or independent researchers to come and study natural history specimens, and to truly understand the unique life forms in India. We are at the crossroads for biodiversity in India. We are losing many species. We really need a modern a national institute where we dedicate everything for biodiversity science, and it connects taxonomy, ecology, evolution with technology and open access. So I work here, I head the research collections at NCBS, and this place is built with that framework in mind. It is an integrated research collection, and it combines all of this that I have been talking about since the past 10 minutes. And it functions simultaneously as a research infrastructure for scientists, as well as educational platform for public engagement. Thank you so much.
Thank you. Uh, the floor is now open for questions. If you have any questions, please go ahead. Yeah.
Hi, Pritha. Thank you for that lovely talk. And you emphasized a lot on the potential of whatever, even the dead birds that we see around, is the potential of the research platforms that lie ahead of us. So I think I'm just curious, like, if is there any way in which people, because all these people are bird watchers and they come across a lot of dead birds or other dead animals from across other taxes, do you have is there any way in which this potential dead specimens can be of use and can be curated in the collections, and what that protocol might be?
Yes, so yes, yeah, that can be done. For example, I was to talk about feather library. We only focus on dead birds, and it has to go through a screening through the rescue centers, and then only can we take it. Because if it has died of some disease, we usually don't take those. But the quality control is important. But again. Uh, just to emphasize, like whatever is dead outside we did not collect, right? It's only for the birds I mentioned, because it's an important resource, and you cannot collect, you're not allowed to collect bird specimens, otherwise you don't get permissions from the forest department. But for other taxa, for many insects and all, only if you know that what the dead specimen that you're finding is could be useful for something, can you collect it. Because if an insect is lying dead somewhere, it might be food for some other animal also. We need not collect everything that we see as dead animals. But and yes, you can just write into us, and we can guide you how to deposit it to our collection. Deposition is absolutely free and open. You just have to write to us, and we need some paperwork. We do a lot of paperwork because we need that trail to be there so that every information is traceable. So but it's quite easily done. Just contact me if you find anything and you want to deposit with us, or yeah.
Ma'am, one doubt. Do we have any modern ways to isolate genes from the formalin deposited specimens? I mean, formalin preserved specimen.
Yes, so very good question. So we do not encourage deposition in formalin because of the reasons you're asking the question. It's not easy to extract DNA from those. So we use only 70% alcohol to preserve our specimens, and if we, yes, from 70% alcohol you can, but from formalin it's a little difficult. There are methods people have tried with formalin preserved specimens, but it's difficult. You don't get intact sequences from there.
Okay, thank you. Then another one question. This NCBS is having the permission to collect. I mean, the National Biodiversity Authority of India is it a National Biodiversity Authority of India recognized repository?
NBA recognized repository? No. So NBA only recognizes national repositories, which are few. No, NCBS is an institutional repository. It's not a NBA recognized.
So we cannot deposit that type specimens there right now.
You can. We have a lot of type specimens.
Yeah, but as per the NBAI, they are not allowing us to deposit the holotypes or paratypes in the non recognized repository. That's why I asked, because recent some of the papers I have seen NCBS, they have submitted the specimen, but
Yeah.
NBA is not asking us to submit there. That's why I asked.
I mean, you can always deposit the holotype in any of the NBA recognized institutes, and you deposit the paratypes with us. That can also be done.
Okay, thank you.
Hey Preeta, can you tell to the audience a little more about how different is Feather Library specimens compared to the traditional specimens in other museums?
Okay. Okay, so Feather Library is built upon this philosophy of, that the dead birds don't go to waste. We do find a lot of dead birds around, and if this could be useful for research, so that's the ideal on which Feather Library was built. In other traditional museum collections, a lot of the older collections were a resultant of hunting, if I'm not completely wrong. Some hunted specimens were there, but nowadays it's not. Yeah, so
The way you are preserving specimens, spread wing, other
oh,
tissues,
that way.
those
Okay.
things
I thought about the philosophy. Okay, no. So yeah, so we have two kinds of preservation. So one is like we have the entire specimen spread for morphometry and all of those parts, and also we are documenting individual feathers. So that's why it's called a feather library. So all the so you have like a collection of just the feathers, like the different kinds of feathers found in a bird, and also you can compare it with an intact specimen. So and also you have tissues kept separately so that you don't have to harm the specimen. Maybe 10 years later, if you want a tissue, we also have a tissue repository. So we try have a base for comparison, which are the intact specimens, and you have the tissues and the feathers and other things kept separately. Is it? Okay.
So, do you think that this existing collections in the, mostly done in arsenic, for example, what we have in Trivandrum Natural History Museum, most of them are in arsenic preserved. So, do you think that those kind of things can be networked or can be pulled into the new system that NCBS is building up, so that it can be more and more useful when you're looking at the tracing history, evolutionary history, and those kind of things. That's one thing.
Yes.
And how the feathers are being preserved. I still remember I got a lot of feathers in my fieldwork days, but that time NCBS didn't have this, for the museum. So, yeah, just asking about how the feathers are being preserved.
Yes. So I think there are two parts to this question. One is the arsenic preserved specimens. So I'll just emphasize here that none of the specimens in our collection has any chemical associated with it. We maintain a climate controlled facility, which is unique among Indian collections. So we do not use any kind of preservatives, but we do right now we do not have a existing network with other collections to build this network, but this is something that can be done. And because feather library is also new, it's about 3 years old. So I think this is a good idea, and we should think in that line to have a network of all these collections, which we want to do in the future. And the feathers are preserved as in they are individually marked as the kind of feathers. It's not that we collect feathers, we collect feathers from the birds. So we have specimens from which the feathers are extracted, actually, like pulled out and preserved. We do not really collect feathers and put them in the collection. So it's taken out from the bird specimen, like from one wing, the other wing is kept intact and spread out, so that, as you can maybe see in this, yeah, so most of our specimens are kept like this. You have maybe one wing has all the feathers taken out to make the repository, as you can digitized repository, as you can see in the bottom photos, and the intact specimen has the wings spread out, so that on the other wing is spread out, so that you can compare like what feathers form which part of the wing. So yeah. So the feathers are coming from the specimens and not.
Okay, not from the. So there is actually a possibility that we all encounter feathers here and there in non-forest areas and many places.
Yes.
So I'm just thinking, like, will that be a legal issue if we go for those kind of things are being collected for a scientific purpose in India? That's Praveen. I'm just also asking about the kind of policy thing that should come out from this meeting. So maybe it is as available in many places, like birds always shed and somehow.
Feathers, yes.
Suppose a raptor catches a small frugivorous bird, definitely you will get only the feathers on ground. So there is a possibility that these all can be pulled into the museum without much of an effort, with a set of protocols and preservation and transporting mechanisms. So that can be a good thing in future.
I think that's a great dimension to expand. Like I said, that feather library is new, and I think it aims for expanding in different dimensions, and this could be one, because we wanted to have this as a very legal framework. That's why we need the permission from the forest department, the rescue centers, and then it comes to the collection, so that there is no conflict at any point in time. But maybe this is a good direction to think about. Yeah,
because these specimens are available
Yes.
even in forest area, non-forest area, birds are everywhere.
yes.
So you cannot limit like, okay, this is only forest property. Birds are across the continents. So we should really look into that frame when you talk about birds.
Yes, the permits that we have are actually state-wise. So we also have a campus bird hit project. We get birds from which collide with the windows, and we collect them also. So it's a state-wide permits. It's only for 2 states right now, but I think we would be expanding.
Just to add, the challenge is to ensure that the bird is dead. That's the
Yes.
reason why this protocol is there.
Yes,
Because the
yes.
moment live birds are there, policies change.
Yes. So, like, people can ask, where did you get this feather? Did you pluck it from a live bird or not? Or did you find it on the ground? There's no way to verify that. Yes, yes. So, which is why we have this proper channel as of now. Yeah.
Any other questions?
So I have okay.
Please sit down.
Yeah. So I have a question on establishing new species based on the museum.
Yeah.
So say I were to find unrecognizable butterfly in the wild, or birds also count, but it's easier with butterflies. So I wasn't able to ideate based on a field guide or anything. So how exactly would I like establish which species it is, or if it's a new species? Would I have to collect the specimen, or is there any way to, let it live?
Yeah, good question. We are always at that conjuncture. You know, when we are doing taxonomy, we think of, okay, whether to let it live or, possibly this is a new species. So taxonomists think like that. So when you encounter something, first is if you are not, like, I'm talking about you maybe. So if you, I don't know how, like qualified you are as a taxonomist. Maybe you are just a nature enthusiast. I'm just saying. Okay, so if you are just a nature enthusiast and you have not experienced this species in any of your past whatever adventures out there, and you feel that this could be something new, first is photo documentation. And then you contact with an expert in the field. There are established taxonomists for all the taxa. Somebody who specializes in a butterfly, you send that photo to that person, that this is what I found here, could this be a new species? And when there is some doubt that okay, it could be a new species, you have to go back and then collect the specimens. But if you are a qualified taxonomist yourself, and and you think this could be a new species, if you have the proper permits and your ethics is in place, so you can collect the specimens. So it all depends on how well versed you are. So I am a trained taxonomist and an ecologist, and if I have permits to collect specimen from a particular place. For example, if I see a moth, I'm a moth biologist. If I see a moth in Wayanad, which I think could be a new species, I wouldn't just collect it. I would take a photo. I would ponder about it for a while, then come back with proper permissions and try look for a specimen. So it comes with a lot of responsibility as well.
Okay, thank you.
Yeah.
These days, you don't really have to look for an expert. You can just upload it in INAT if it's any specimen or eBird, and there are other people who will help you to identify it.
Yeah, but then for insects in general, it's not as resolved as for birds maybe. So yeah, so the INAT algorithm also works on how well other specimens are identified.
But for a person who is
Yes,
new,
yes.
this may
That's
be the
an
first
easy
step,
way to verify.
and there may be
yes.
some experts who can take it up.
Yes, definitely.
Excuse me, you were talking about that through the feathers, when zoom in, you can identify that that species adaptation and evolution, those kind of things. If we got the two feathers of same species of different individuals, is there any similarities or is there any unique features among them or between them?
I'm not a bird expert at all, but I'm sure because feather microstructures, all the ultrastructures through scanning electron microscope has been used to understand flight dynamics and other kind of adaptations. This can be done, but I think I'm not the right person to answer at this resolution. But this is something that is done usually to compare feather microstructures between species or with the same species, different individuals. So it is a common practice. You can do it.
Maybe I can just add one more point. In aircraft strikes, the aircraft gets damaged by birds hitting it, and they don't know which species it is. So they just collect this specimen, the feathers. They have done the microstructure studies, and based on that, they're able to identify the species or at least the group from which it comes to. So to that extent, I think feathers can be unique.
Yes.
Hi. So do you also do other like molecular biology studies or like DNA barcoding or things like that to the specimens that you get?
Yes. Yes. So it all, like I was showing you about the geckos. That team from Thakre Wildlife Foundation, they usually keep the tissues separately for molecular work, or they have already done the molecular work and then they give the specimens to us. But yes, it depends on who is doing what kind of research. So we have like for birds also, we have the tissues kept separately so that molecular studies can be done as well.
Any other questions? So thank you. Thank you.
Thank you so much.
Just I wanted to add one more point to what has been said. We have been talking more about how collections are useful for us for biological studies and things like that. But beyond that, for humans also, these collections are important. For instance, seed banks are very important for future food security. And I was just reading a book fairly recently called The Secret Garden, The Forbidden Garden in Leningrad. Talks about how these scientists, Vavilov and his colleagues, have protected specimens from being taken over by Germany because food was scarce, because this city was under siege for several months and people were dying because of hunger. But these people who were responsible for it had tons of specimens, different kinds of potatoes, different kinds of other grains collected by Vavilov for last 30-40 years. And they protected it throughout the siege and made sure that those specimens were available. And now, of course, we have Slavbard and other places where in Norway, where all these specimens are protected and kept for future use. So I think that is another area. For health studies also, some of the, you can take DNA from this, of the viruses, and you can understand how diseases were there in different bird species in the past. So I think it's a very useful thing to have biological collections, and I hope we appreciate and use these collections wisely. So I think we'll now move to the next speaker. The person who's going to speak is Parvati, and she's going to talk about using archives for ornithological research. Parvati is an archivist at the archives of NCBS, where she organizes and catalogs collections to make them publicly accessible. With a background in environmental history, she has archived materials that document the complex relationship between communities and their environment, as well as narratives of environmental activism. Her work extends beyond archiving to active research on these collections, identifying connections and curating research guides that help scholars and the public navigate and engage with the archives.